Hi everyone, I'm doing my presentation on a mutation of the RHBDF2 gene, and it's an autosomal dominant mutation that is associated with tylosis with esophageal cancer. The RHBDF2 gene is officially called rhomboid 5 homologue 2. It is located on the long arm or the Q arm of chromosome 17 and has 21 exons. The exact position of this gene is 17q25.1 and is indicated by the red arrow seen in the image here. The RHBDF2 gene codes for a highly conserved 7-transmembrane rhomboid protein that is found on the endoplasmic reticulum of cells as seen in the image here on this slide. The RHBDF2 protein acts as a serine protease and is linked with the epidermal growth factor receptor and therefore the epidermal growth factor pathway. RHBDF2 is believed to be linked to the preparation of epidermal growth factor proteins and is believed to act by cleaving bonds of the epidermal growth factor proteins. The RHBDF2 gene also plays a role in the secretion of tumor necrosis factor alpha, which is an inflammatory cytokine that produces monophages and macrophages. TNF-alpha also triggers cell signaling processes that result in apoptosis or necrosis of the cell. TNF-alpha is crucial for resistance against infectious diseases and cancers. The mutation of the RHBDF2 gene that causes cancer is a result of a missense point mutation in exon 6 of the gene. This mutation exchanges an isoleucine for a threonine, and it appears to be a mutation of the second nucleotide of the codon in which a uracil is exchanged for a cytosine. This results in an autosomal dominant mutation that is found in a heterozygous fashion with all affected individuals. This mutation results in a gain of function of the EGFR signaling, thereby activating the EGFR pathway. The mechanism, however, is not well understood as this is a rare genetic mutation and therefore a rare cancer, making it difficult to study. It is believed that by activating the EGFR pathway, the RHBDF2 missense mutation also activates the downstream signaling event of the EGFR pathway, causing secretion of specific EGFR receptor ligands, causing growth to occur in an accelerated fashion, which results in the formation of tumors. This missense mutation of the RHBDF2 gene causes tylosis with esophageal cancer. As mentioned earlier, this is an autosomal dominant gene mutation. It is associated with thickening of the skin, especially in the palms of the hands and the soles of the feet, which is seen in Figure A. It is also linked with an oral leukoplakia, which is the thickening of the oral mucosa seen in Figure B. Lastly, this gene mutation is associated with esophageal squamous cell cancer. This mutation is relatively rare, but has been seen in pedigrees in the US, Europe, and China. As of 2012, esophageal cancer, largely of the squamous cell variety, was the 6th leading cause of cancer-related deaths, and while esophageal cancer due to RHBDF2 missense mutations only accounts for a very small fraction of these cancers, it is hoped that by better understanding esophageal squamous cell cancer caused by RHBDF2 gene mutations, researchers will be able to better treat all types of squamous cell esophageal cancers. Currently, treatment for tylosis with esophageal cancer caused by this mutation is focused on lifestyle changes and early detection rather than targeted gene therapies. The missense mutation of the RHBDF2 gene is also associated with rapid wound healing. As such, using tissues that express this mutation is under investigation as a possible therapy for other disorders associated with slow wound healing. On the other end of the spectrum, high levels of RHBDF2 gene expression is associated with kidney renal clear cell carcinoma, and therapies to silence or downregulate the RHBDF2 gene are under investigation to treat this cancer. As discussed previously, the RHBDF2 gene plays a role in inflammatory processes due to its role in the secretion of the tumor necrosis factor alpha. As such, this gene is under investigation as a target for treatments related to inflammatory bowel diseases, such as Crohn's disease and ulcerative colitis. It is also a target for treating inflammatory arthritis and other diseases associated with tumor necrosis factor alpha. And this is because RHBDF2 could play a role in the overexpression of an individual's inflammatory responses. Lastly, here is just a list of all the references I used to put together this presentation. Thank you for watching.